Hey guys, welcome to the first section of the microbiology final review. So basically, what I do is I have like a giant slide full of chapters like 1 through 10, and then 11 through whatever the rest of them are. However, I think it does skip over all the pathogens, over viruses a bit, over multicellular parasites, all of that, just because, like I said, like in my past reviews, that's all about memorization. Like, I really can't help you much with that. Um, but in addition to that, I do have some review questions that I prepared. And it's kind of just in a slide format. So it'll have the slide and then you'll have the answer underneath to kind of help you with that so that you can ask yourself the question and then answer it below. I just don't like to give them out earlier in the semester, but this is more for review. So this review I'm hoping won't be super, super long. There are about 30 something slides in it, but it's all review from what you know so far. And I hope this really helps. I know that we had issues with my other review, so this is the re-recording of it. I'm really hoping it'll still be as good. But if you have any questions, feel free to reach out to me. My schedule has picked up a little bit, so I might be a little bit slower on responding to you, but I'll respond as fast as I can. So with that being said, I guess we should begin. So the first thing that we talked about was the history of microbiology and many important people that were very influential in developing microbiology. People like Edward Jenner, Louis Pasteur, Rudolf Virchow, Robert Koch, Paul Ehrlich, all of those. So on my slide, I've got the list of their names, and then it just asks you, like in a question underneath, it's like, what did all of these people discover or develop? And so if you can really nail those down, if you have flashcards for those, awesome. I'll send this out so it's a little bit easier for you because it's easier for, at least for me to learn. It's easier for me to study like things that I would have to memorize myself rather than having someone just speak at me on it. But if you can remember like the main people of microbiology and kind of what they studied, you should be you should be really good on that one. The next thing we went into was like a brief chemistry lesson. So the elements in all living things. I told you guys to remember CHNOPS. So carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfate. Those are kind of the building blocks for every molecule in the human body or in any living being. On top of that, just remember that the basic building blocks of everything are atoms. You have I don't know where I was going with that. And the charged atoms are ions. So if you have a positive ion or a negative ion, so you'll have an anion or a cation, that's just kind of what that means, is that they have a positive or negative charge associated with them. And then bond types, we've got ionic, covalent, and hydrogen. Um, and just kind of, I think, understand the difference between those would be really good. So like ionic would be kind of like rubbing a balloon on your head and then being able to stick it on the wall. It's that attraction between charges or between ions. And then covalent, they end up sharing electrons. They kind of like link together. Their octets link together. And then you've got hydrogen bonding, which is something that we see a lot in nucleic acids, and it's super important to remember and understand. Just because hydrogen bonds are fairly weak, but when you have multiple hydrogen bonds like within the same molecule, it makes that bond so much stronger. It's like taking one piece of spaghetti and breaking it versus taking like 12 pieces of spaghetti and breaking it. You're going to have a lot more resistance between those 12 pieces than you will with the one. And that's like the best comparison I can give for hydrogen bonding. Next we talked about metabolism. So we talked about like catabolic processes and anabolic processes. And the best way I could remember this was catabolic reminds me of catastrophe, and anabolic, I thought of anabolic steroids, and when people do steroids, they get like super big and jacked and whatever other terminology you want to use. So with catabolic, it's just breaking down things. So like with catastrophe, if there's a hurricane or a tornado, it's going to break down everything. and it's considered to be exergonic, meaning that it releases energy. And typically this is done through heat, but it's each bond that it breaks will release energy. And an example that we gave of this was hydrolysis. I feel like you guys understand hydrolysis really well. It's just taking molecule like C and breaking it down into A and B and then water. You're adding water to molecule C to get molecules A and B. And then with anabolic, what you're doing is you're building or synthesizing new molecules. So this is considered to be endergonic, meaning you have to put energy into it in order for the reaction to occur. An example we gave of this was dehydration synthesis, which is just the reverse of hydrolysis. So you'll take molecule A and B, and to add them together, you will get molecule C in the end plus water. It's going to take that water out of it. We see this a lot with protein synthesis and with nucleic acids. But we don't learn too much about that in this class. Next we talked about pH. So our neutral is going to be our pH of 7. And then for each one that you, like each difference, so like 0 to 1, you have 10 times the amount of, I want to say it's OH because it'll go up in OH and it'll go down with H+. So acidic is going to be anything above or below 7, and basic is going to be anything above 7. And the normal range for most organisms is between 6.5 to 8.5. And if you notice, that's close to the neutral range. Our bodies function a lot better at that neutral range, not so much as at the acidic range. Different parts of our body can, for example, the vagina, stomach, intestines, I think, they all have a slightly lower pH than like 6.5. Like stomach acid is anywhere between, I think, 3 and 4. But for the majority of our body, the pH is close, like, to that range of 7 plus or minus, like, 1.5 or whatever. So next we talked about organic compounds. In my slides that I'm going to send out, there is a link, and I want you guys to watch it. If you watched it earlier in the semester, awesome. It's the Amoeba Sisters, like, video that I did, that I had sent out. I really like that one. I think it explains organic compounds a heck of a lot better than I can, but I am going to go into organic compounds just a little bit. So our organic compounds we have are carbohydrates, lipids, proteins, and nucleic acids, which would be your DNA or RNA. Either one falls underneath that. And I'm going to go over each of them individually, but I really, really, really do suggest watching this video. I think it helps a lot. I know it helped me a lot with understanding everything for the class. So again, that will be linked below in my slides for you. So carbohydrates, the ratio for them is a 1 to 2 to 1. So carbon has for every 1 carbon, you have 2 hydrogens and 1 oxygen. And I mean, the biggest thing I can remember is, like, in high school, we would talk about C6H12O6, and you talk about it all the time. Like, we talked about it when we started talking about aerobic respiration and everything. You see that characteristic of the 1 to 2 to 1 with the 6-12-6, like it's 1 2 1. Anyways, so you've got mono, di, and poly carbohydrates or saccharides. Saccharide is just another name for a sugar, and they're super important because they're used to make glucose, which is the body's main energy source. And you can end up getting glucose by breaking down, honestly, any series of mono, di, and polysaccharides, or di and polysaccharides. But di just refers to 2 saccharides being linked together, and poly refers to many. I don't think she gave a specific number in this class, so we're gonna just assume more than 2. But carbohydrates are just the basic, like, energy source for all of life. That's why plants make it, and we eat plants. Like, we get our energy from that because we're not able to produce that sugar ourselves. We can break it down from different polysaccharides, but we can't make glucose ourselves. The next one we talked about was lipids. So lipids is the only one that's not a polymer, meaning it doesn't have multiple, like, components. So, like, with saccharides, you'll have one saccharide bonded to another saccharide bonded to another one. Lipids don't do that. Lipids instead just they can't have different structures, like I'll get to in a second, but they're the only ones that aren't a polymer. But just like carbohydrates, they are made out of carbon, hydrogen, and oxygen. So if she asked a question about, like, which classes of organic compounds are made of just carbon, hydrogen, and oxygen, you could put carbohydrates and lipids, because those are the only ones. However, it does get a little complicated when we get into, like, different types of lipids. But for the most part, they're made of carbon, hydrogen, and oxygen. So lipids do form cell membranes in the form of phospholipids, and they do store energy. You're going to store a lot more energy in lipids than you will in carbohydrates, because carbohydrates are readily broke down. So then we get into simple lipids, complex lipids, and steroids. So one thing that I thought was really cool is a triglyceride is basically just a lipid attached to, in essence, 3 different saccharides, which I thought was really cool, because what it does is it kind of locks up those sugars so they can be used at a later time. So, like, when you eat too much, and everyone's like, well, if you eat too much, then you're going to gain weight. Yeah, because that's what this is. You're not able to process all of that glucose at the time, so it'll store it in the form of triglycerides and fats so that you can then use it at a later time. The other ones we had was complex lipids. So these ones do contain carbon, hydrogen, and oxygen, but they also can contain phosphorus, nitrogen, and sulfur. And examples would be phospholipids, but also cell walls found in Mycobacterium. And we talked about Mycobacterium, but that's another place that'll contain these type of complex lipids. And then the last one would be steroids, and the example she gave for that was cholesterol. I'm trying to think I think that's good on lipids. Proteins. So these ones are always going to contain carbon, hydrogen, oxygen, and nitrogen, and sometimes sulfur. It kind of depends on whatever structure we end up using as the side chain, because they're going to have the same, like, basic structure with that carbon, hydrogen, and oxygen, as well as the nitrogen, but they have different side chains which give you different amino acids, which is where we can have this sometimes S thing coming in. And if you ever take organic chemistry, you'll learn about it. If you take biochemistry, you'll learn about it. But so this is the most common macromolecule in cells. It's used as an enzyme. It's used as, like, transport channels. It's used as exotoxins, bacteriocins. Like, it's used for literally everything. But they do perform numerous vital functions in living things. They catalyze reactions so that, like, things can go to completion. We can make whatever product we need to make. They are formed by amino acids linking together. And then we've got structures. So I've talked about this before, and everyone seems to laugh about it, but this is the best way I can describe it. So there's primary, secondary, tertiary, and quaternary structures. With primary, the best thing I had this teacher in high school when I took AP Bio, and she would have us do this really kind of stupid thing, but it's always stuck with me, where she would yell primary structure, and all of us in class would go like this, and we'd stick our arms out, because primary structure is just a chain of amino acids. Then she would yell secondary structure, and we'd try to form, like, a beta pleated sheet or an alpha helix with our hands. Then she'd yell tertiary structure, and we'd all, like, duck onto the ground and curl into a ball. And then she'd yell quaternary structure, and we'd all, like, scoot closer together underneath the table to form, like, a glob. So with primary structure, it's going to just be that straight chain of amino acids. Secondary structure is where it kind of starts taking form, where it's going to form either a beta pleated sheet, which kind of just looks like zigzags, which is why I did this thing, or it'll form an alpha helix like we see in our DNA. Then we've got tertiary structure, and that's just kind of, it starts to condense almost and like glob in on itself, so it looks like a giant mixed of glob. It's a glob. And then with quaternary structures, you have multiple tertiary structures coming together to form that protein. And when they do that, that's when we can find active sites. That's when we can find like a fully activated protein. So, kind of a lot. But I feel like that's something that you guys understand really, really well. And then what can denature a protein? We kind of talked about this. High pH or extreme changes in pH, high temperatures, and salt concentration can actually denature a protein just because it's not able to work at the level it needs to, or it'll start breaking the bonds within like that protein so that they can't really form the way that they need to in order to complete whatever reaction or in order to do the function that they were intended to do. So then we have nucleic acids. So this includes our DNA and our RNA, and this one contains carbon, hydrogen, nitrogen, oxygen, and phosphorus. And I feel like once you guys kind of understand this, like once you think about the structure, you'll understand it a little bit better. So with nucleic acid, they've got a sugar phosphate base, right? And sugars, like we talked about, are carbohydrates, which have carbon, hydrogen, and oxygen. So that's a no-brainer. And then it talks about how it's got a phosphate as well. So that gives us our phosphorus. And then we have nitrogen-containing bases. So that's kind of like an easy way to remember what elements are in nucleic acids. We've got the carbon, hydrogen, nitrogen, oxygen, and phosphorus. And you can see that all if you look at the structure of the DNA. It's formed by nucleotides linking together, and so those are the basic building blocks of your nucleic acids. Nucleic acids are used for making proteins, they're used for heredity, genetic material, and then this is going to be your template for protein synthesis. So basically what happens is you convert your DNA into RNA, which can then be read and turned into a protein. In your DNA, one thing to note is that there's always 2 hydrogen bonds between A and T and 3 between C and G. And the best way that I could remember that is if you look at a T, it's got like 2 lines, and if you look at an A, it's got like 2 lines. Just don't worry about like the little one in the middle. So that's how I remember that. And then C and G, if you put a C on top of a G, it makes a backwards 3. Kind of out of the box, kind of weird, but that's the best way I could just remember that one. So like kind of just a brief review of that. It's important to remember what like the basic building blocks of each macromolecule are. So if I were to ask you what the basic building block of a carbohydrate was, I would want you to say saccharide. If I were to ask you what the basic building block of a protein was, I would want you to say amino acid. And if I were to ask you what the basic building block of a nucleic acid was, I'd want you to say nucleotide. If you guys can differentiate between those two, you should be golden on those. Or between those like 3, yeah, you guys should be golden. We didn't really talk about that. Okay, so enzymes. With the naming of enzymes, they're usually going to end in ACE. So if you see something that ends with ACE, you should be okay to assume it's an enzyme. Examples would be lipase, sucrase, oxidase, decarboxylase. And the different components of it is you've got your apoenzyme and your coenzyme. So your apoenzyme is going to be your protein part that I talked about. It's going to have the binding site. It's going to be made entirely of proteins, and often it's going to be inactive. Then you've got your coenzyme, which is your non-protein part. It's often a metal ion, which can come in and then activate it, and that's going to be what, like, that coenzyme is going to be what activates that enzyme so that it can start proceeding with the reaction. And another name for like the two of them put together instead of just enzyme is called a holoenzyme, which basically just means whole enzyme. Then we talked a little bit about enzyme inhibition. Basically, a molecule binds to an enzyme to decrease its activity. So we talked about competitive versus non-competitive enzyme inhibitors. The best way I could remember this is like in competitive, I always thought of like sports and football and competing for like a goal, or competing like in basketball, competing for the ball. So what happens is these enzyme inhibitors will compete with the substrate for the binding site. So if that enzyme inhibitor reaches the active site before the substrate can, it's going to make it so that substrate can't bind to the active site. Whereas non-competitive, I used to think of stuff like, I don't know, like school. Like you had to be more smart about it. So what's going to happen is you've got like your whole enzyme, right? Your binding site would probably normally be up here. But with non-competitive, they're going to come bind at a different site of that enzyme, which will essentially change the binding site so that they can't, so that whatever substrate is normally there can't bind in the active site, because the active site is a different, it's a different shape now. And a lot of times it's caused by removal of that cofactor that we talked about, like that ion. It'll just make it so that active site just isn't active anymore. It doesn't work. The substrate won't fit in there. It's like trying to mash two puzzle pieces together that just don't work. But that's competitive enzyme inhibitors versus non-competitive enzyme inhibitors. And I have a little picture on like the slide that you guys can look at. And like when I send out these slides, you are more than welcome to follow along with me talking and look at these slides. I think it might help you a lot more if you do that. But we're making great time on this. So then she talked a little bit about phagocytosis, pinocytosis, and receptor-mediated endocytosis. So with that being said, endocytosis is a eukaryotic function only. And the way I could remember it is eukaryotic and endo start with E's. So that's one way to kind of like remember the difference between them, or remember that they are the same, like only done by eukaryotes. But phagocytosis is going to be eating. Pinocytosis is drinking. During one of my reviews, one girl said it's like drinking a pint. And I feel like that was a really, really good way to remember that. So I am definitely sticking with that. But, so phagocytosis is the cell is going to basically like Pac-Man and eat whatever like bacteria or molecules there are to bring them into the cell. Pinocytosis is cell drinking. So it's going to end up bringing nutrients, extracellular fluid, possibly ions into the cell just because it needs them at that time. The other one is receptor-mediated endocytosis. And so you're going to have like your normal receptor, right? Or your normal cell membrane, but it's going to have like little proteins on the outside of it. And so when a ligand comes and binds to them, once all those are filled, it can start forming a vesicle and like bringing it in through the membrane to form like, it'll have like a cell membrane, and it'll have receptors, and then it'll have whatever ligands were needed, and it just brings it into the cell. I thought this one was really cool, and you kind of see it a little bit when we talked about, or like a form of it when we talked about the immune system, but this one is just receptors that are waiting for whatever it needs to be binded to it so it can bring it into the cell. Next, so passive versus active transport. This is something that I feel like is very, very, very important to differentiate between. And it's because like once we start talking about like aerobic respiration and understanding the difference between like it's important because it has to do with ATP. But so passive transport molecules are essentially free to cross the membrane. So an example of this would be like osmosis, where water will just cross wherever it needs to establish an equilibrium or whatever osmotic pressure is needed. And it's going to follow like a concentration gradient, right? So this will happen in osmosis, facilitated diffusion, and with small non-polar molecules. If they're polar, they end up kind of getting caught within the cell membrane, or they just won't pass through at all. And like the proteins that allow for some of these molecules to pass through, they're always going to be called channels. Channels kind of insinuate that it's a passive process. And then we've got active transport. So for active transport, ATP is needed to push ions or whatever is needed against the concentration gradient. So instead of it going from a high to a low concentration, it's going to go from a low to a high concentration. And these are always going to be called pumps because it takes energy to like run a pump or to pump something across. And so it's needed in order for you to pump whatever like ion is needed into wherever it needs to be, whether it's outside of the cell or inside of the cell. Okay, so now we start getting into the difference between eukaryotes and prokaryotes, which honestly I think is probably one of my favorite, and maybe that's just because I've learned about it since like 7th grade, but eukaryotes have a nucleus, they have organelles, they contain histones, and if you remember, histones are kind of what helps cause, it's that protein that helps cause DNA to be able to condense on itself and helps wrap it up so that it's contained in a smaller unit. They only have a plasma membrane, so they're only going to have that one phospholipid bilayer, and then they reproduce by mitosis and meiosis. And I feel like you guys understand mitosis and meiosis, not that you need to for this class all that much, but yeah, it uses mitosis and meiosis. In contrast, prokaryotes, they're not going to have a nucleus, but they're going to have a nucleoid region. So it's like one area of the cell where the DNA is contained. They also have the HU protein or the HU protein, which causes supercoiling. And the best way to describe this is like if you have ever used one of those phones that have a cord on it and you like accidentally wrap like you're talking on the phone and you like turn the phone a little bit and then put it down, you'll see the cord of the phone will like wrap a little bit. That's what we call supercoiling. So prokaryotes are going to have supercoiled DNA and plasmids. And plasmids are just a circular form of DNA. They're not going to contain any organelles, and they reproduce by binary fission. And they have a cell wall, which is another thing. Because we're not going to have a cell wall in eukaryotes. It doesn't work out very well. And if you think about it, like if your cells had a cell wall, your body would be a lot more rigid and it would be a lot more different. Like if you look at the stem of a plant compared to your skin, that's the best way I've always thought about it, is like the stem of a plant is very rigid and it doesn't bend as often, and especially with where we're larger organisms, we'd probably end up looking more like a tree if we had cell walls. Next thing, just like a little brief overview, is we've got cell organelles. So I tried to come up with some really, really good ways to remember these. And they work for me, but they might not work for you. So I would suggest making up some for yourself. So the rough ER versus the smooth ER. With the rough ER, it's going to have ribosomes embedded in within the membranes of the rough ER, which is why it's rough. And then you've got the smooth ER, which is smooth because there's no bumps from the ribosomes. So rough ER is going to produce proteins and phospholipids, whereas the smooth ER is going to produce steroids and phospholipids. So the endoplasmic reticulum, or the ER, is going to produce phospholipids either way, Steroids and smooth start with S, so that's how I remembered that steroids went to that. And then it was just kind of remembering that proteins are made from, like, ribosomes help make proteins, and that's how I remembered that. And then we've got lysosomes. So those are organelles that contain digestive enzymes and digest worn out food, cells, bacteria, viruses. I kind of thought about this as like the sorry. I thought about this one as like the city dump, because it's where things go to be broken down and digested, quote unquote. So I just thought of lysosomes as the as the dump, or the city dump, or the garbage man coming to pick everything up and start breaking it down. Nucleus I always thought of as like city hall or a blueprint. So because it's found only in eukaryotes and it holds all of the DNA inside of it. And then ribosomes, they're found in both cell types and they're involved in protein synthesis. I didn't have a good way to remember ribosomes. Man, I am so sorry. And then we've got the Golgi body, which is just a series of membranes. It's located right next to the endoplasmic reticulums. It helps package molecules and send them where they need to go, and it can modify them if needed. The best way I could remember this one was it was like UPS or FedEx or Amazon Prime, whatever you want to call it. Basically, it just kind of packages things and sends them where they need to go. And I think, yeah, that's all I had for cell organelles. If you guys have any questions on those, please let me know. Or if you have a better way to remember them by giving them different jobs, like I said, like the city dump or a garbage man and like UPS, I would be more than happy to hear from you on that, because I need a better way to remember and teach that. The next thing we talked about was bacterial cell shapes and arrangements. So she doesn't talk, like, it's on the slide, but she doesn't often talk much about diplo versus tetrad versus sarcinae. But just so you know, diplo is 2, tetrad is 4 cells in one layer, and then 8 is 2 layers of 4, so 2 tetrads on top of each other, giving us 8 total cells. But for bacterial cell shapes and arrangements, this is something you do need to know. So if you have coccus, that's going to be circular, and the way I remember that is there's 3 C's and an O in that, and they all look like circles all in a row. So that's how I remember that one. Bacilli is rod-shaped, and the way I remember that was there was an ILLI, which all look like rods. And then we've got spiral, which would be like your spirillum, which looks like corkscrews. And I feel like that one's super self-explanatory. But then we've got 2 different arrangements that they can be in. You can have staph or you can have strep. And with staph, that's going to be a cluster. And with strep, that's going to be a chain or a line. And the best way I can describe that one, when I was younger, my mom would tell me I used to get strep throat all the time. And so my mom would be like, "Oh, you have strep throat." And I thought that she said stripe throat. So I would go check the back of my throat for little lines on the back of my throat, thinking that I had like tiger stripes in the back of my throat. I was really disappointed when I found out that it didn't look like that, but that's the best way I can think of to remember strep. And then staph is just kind of remembering that it's going to be a cluster. And then to like mix them together, so like let's say you've got a bacterial colony that is both coccus and staph, it would be staphylococcus. You're going to add the YLO in between that, or Staphylobacilli. If you've got like a long chain, it would be Streptococcus or Streptobacilli. On strep, you just add a TO. So I feel like that one's pretty good too. bacterial cell shapes are super cool, and it helps you with, like, identifying so much. Okay, so now we start getting into a little bit tougher stuff, so like gram staining. So with the gram staining technique, we've got a primary stain, we have a mordant, a decolorizer, and a secondary stain. Your primary stain is going to be your crystal violet. Sorry. It's going to be crystal violet because it's going to start out purple. So both your gram positive and your gram negative will start out purple. Then you're going to add a mordant, which is what we call grams iodine in this case. And then you'll add your decolorizer, which I'm pretty sure is alcohol in this. Yeah, so you're going to add alcohol on this one. And then once you add that alcohol, that's where you're going to start seeing like a difference in color, because you're either going to, because it with those gram positives, you're not going to be able to wash out that purple color, but with your gram negatives you can. And then you've got your secondary stain, which is going to be safranin, which is going to change it to like a pink or a red appearance on only your gram negatives. So after you had finished your gram stain technique, your gram positives are going to be purple and your gram negatives are going to be a reddish pinkish color. And just like a side note on gram negatives, so gram negatives, their cell walls contain peptidoglycan, or bacterial cell walls in general contain peptidoglycan, but gram negative cells have a lot less peptidoglycan than do gram positive cells. And so with that being said, like gram positives have a thicker cell wall. Gram negatives also have an outer membrane. So what happens with like your primary stain is it's going to stain mostly that outer membrane, and then by adding that decolorizer, it'll poke holes in that membrane, allowing that color to wash out of the gram negative, but it's not going to be able to poke as many holes in that really thick cell wall that you'd find in gram positives. And then by adding that secondary stain, you can stain the cell wall on the inside of a gram negative bacteria, which is why we end up seeing that pink. Another note on gram negatives is that there is lipopolysaccharide, which is found in the outer layer of gram negatives, which is what makes gram negatives super, 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 like, toxic and makes us really sick when we get infected with a gram negative, because it contains lipid A, which is something that is very toxic to humans. So there's that on gram negatives, and I have a slide following that shows like the process of gram staining and what each cell will look like on each stage. So then we will talk about glycocalyx and endospores. So a glycocalyx is going to have two parts. It's going to have a slime layer and a capsule. So basically both of them are just to kind of help it evade phagocytosis. So like if a phagocyte comes over and grabs it and it doesn't wrap entirely around it, it can push itself out of that slime layer and like keep squiggling on to wherever it's going. If it does get entirely engulfed, it's got a capsule, which is super, super hard, and digestive enzymes can't typically break it down, so it allows it to continue to survive. And then we've got endospores, which is another like way of surviving. So they're only found in two species, your Clostridium and your Bacillus. If she asks any questions about endospores and what cell types, if there's Clostridium or Bacillus, that's your answer. But basically what happens with an endospore is when it's really hard for a cell to survive in an environment because of increased waste products or extreme changes in pH or not enough nutrients, it'll start essentially shutting itself down and shoving all of its information into a tiny little area of the cell, and it'll form a spore coat, which kind of just surrounds it, and then the outside layer will like get shed off to become an endospore. Now, at this point, they are not living whatsoever. So when conditions are right and they're able to grow again, the best way I can describe it is it's kind of like a seed. Like a plant will make a seed, and then because there's not like, it's not able to continue growing at that point. So, like, that's why you'll get a lot of seeds towards the end of, like, the season or whatever. And that seed, once planted and the conditions are right, can start growing, but it's only going to grow one plant. The same goes for endospores. It's kind of like a seed where conditions aren't right, but as soon as conditions are, it'll essentially reactivate itself and start growing again. But it's not going to ever form more than one cell. One endospore forms one cell. After, like, that one cell is made, it can continue to reproduce. But the important distinction here is that only one cell comes from one endospore. You can't have two cells coming from one endospore. It's not possible. And she has asked questions about that in the past. I missed one of them, and it drove me crazy. She is rewriting the test, so she might not ask a question like that, but just so that you guys do understand the difference, I feel like this is a very important distinction to make. Okay, so we are now starting chapter 7 in my slides. Okay, so chapter 7, we started talking about, like, sterilization, disinfection, degerming, all of that. It's important to understand the definitions for this, and I think that making slides or making flashcards for these would really, really help you guys. So in my slides, the way I have them set up is I feel like anything that is mostly definitions I have on one side, and I've got multiple of them so that you can just go from there and make your own flashcards. I know that end of semester is really, really stressful and you're studying for lots of classes, but this test is not one that I wouldn't, if you've done well all semester, I'm not super nervous about you. Like, I think you'll do okay. If you're not, please take all of my information and all my advice to heart. But I really think if you've made flashcards throughout the year, or throughout the semester, I would just continue using those to help you, like, restudy. But chapter 7. Okay, so sterilization. That's going to get rid of everything. Literally everything. Endospores, viruses, any and all pathogens. They're gone. Disinfection is going to get rid of everything but endospores. So you're still going to end up having endospores there. But you should get rid of, like, the rest of all other cells. Degerming is the process that we use in hand washing. So, like, with this whole COVID thing and everyone being stressed out about it, degerming is key. It's removing germs mechanically. So you need friction and movement to dislodge, which is why, like, when you wash your hands, you should do it for 30 seconds and get all areas of your hands, because it'll end up creating enough friction that it'll rub off as many cells as possible. Then we've got, then we have, hold on 2 seconds. Okay. Then we've got antisepsis, which is the practice of using antiseptics to eliminate the microorganisms that cause disease. This was introduced by Semmelweis. I know you guys have heard that story. Feel free to look it up again if you want to. But it's just using antiseptics within, like, hospitals and being more safe with practices. Then we've got sanitization. So this is removing just enough of the pathogens in order for it to be considered clean or safe. And the example she gave was, like, of toilets that have the little label on it that says sanitize for your protection. So, like, when we clean doorknobs or light switches or anything anywhere, like, within a building, it's sanitized. So it's removing enough of it to be considered safe. It's not going to remove everything, but it's going to be removing as a decent amount, so it's not, like, overrun with bacterial, like, with a bacterial population. So with that being said, we need to talk about what affects the rate of microbial death. So microbial death obviously is killing off bacteria. But what's going to affect it is the number of microbes. So if you have a lot of microbes within an area, at some point they're going to run out of nutrients, or there's going to be a lot of waste products from the bacteria, which makes it so that they can't end up like actually growing as much as they would like, or there's not enough space. Which kind of brings us to number 2, which is environmental influences. This could be anything from pH, temperature, debris, like blood or vomit or feces or whatever. The nature of the suspending medium, depending on like what nutrients are in the environment, depends on what can kind of grow and how long it can grow. Time of exposure, so how long it is exposed to, say, ethanol or other cleaning bacterias or cleaning products. Like with bleach, if you leave it on for like 2 seconds versus 20 seconds, like how much more effective is it going to be in that 2 second, like in that 20 seconds than it was in that 2 seconds. And in addition, like characteristics of the microbe being attacked. So does it have endospore, like can it form endospores? Does it have a glycocalyx? Is it able to be motile? These are important things to consider when you're trying to affect the rate of a microbe's ability to grow, and if you're trying to kill it off faster. So with that being said, the targets of microbial control, we've got 3 of them. So you can either damage the plasma membrane, you can damage the intracellular proteins, or you can damage the nucleic acids. So with the plasma membrane, it's like taking a balloon and poking it with a pin, it's going to pop. So if you tear holes in the membrane, basically all the contents are going to end up leaking out. You're not going to be able to hold everything inside. You're not going to be able to fix any, like you can't fix a balloon that has a water balloon that has a hole in it. You kind of just chuck it away. So if you were targeting the plasma membrane, it's similar to poking a hole in a water balloon and then watching all the water like just kind of leave. Intracellular proteins, you can denature proteins, which essentially stops growth. An example of this would be phenol. If the proteins get denatured or the enzymes get denatured, it can severely slow down the cell processes and make it so that those cells aren't able to grow and able to, like their metabolic pathways may not run as efficiently, or they may not be able to bring in certain nutrients through their membranes. So denaturing proteins is a pretty good way to go. And then nucleic acids. So you can cause not only denaturing of the protein, but you can also cause a change in genetic sequence. For example, with ionizing versus non-ionizing radiation, we talked about how you can either break the DNA entirely, or you can form dimers. So when it talks about like cause genetic changes in the sequence, that can change genetic sequence, forming those dimers would change that genetic sequence. But other examples would be heat. Heat could denature nucleic acids, as well as other chemicals. They're able to like get right in there and kind of cut through the DNA or stop DNA synthesis. So those would be the targets of microbial control. Then we've got physical methods of microbial control. So we've got desiccation, osmotic pressure, low temperature, high temperature. And this is another one that I feel like is a memorization one. So with high temperature, that's going to be boiling or moist heat sterilization. With low temperature, that's going to slow the rate of growth and metabolism. The best way I could think about it is when I'm cold, I don't want to move. Like, if I'm stuck in my bed and it's warm in my bed and I know it's cold outside, I'm not leaving. or if I'm outside and I'm walking, at some point I start walking slower because I'm so cold and my body's shaking that I can't really walk as fast. So low temperature is going to end up slowing the rate of growth and metabolism. Hence why I think that. Osmotic pressure is the pressure which must be applied to a solution to prevent inward flow of water across a semi-permeable membrane. So basically, you're going to end up, this is the one where she had the picture of the fish with all the salt on it. So the salt is going to prevent water from going into the fish, but instead kind of like pull water, in a sense, outwards towards that salt. Then we have desiccation, which would be like making beef jerky. It's extreme drying out. You're going to take as much water as possible out of there because it'll make it so that you end up drying it. It becomes a little strip of leather, like fruit leathers and beef jerky and like dried fruit. That's the best way to describe desiccation. It's an extreme drying out. You're pulling out all the water from it. So using heat, I do have a slide on here that talks about like autoclaving, pasteurization, UHT treated dry heat sterilization. Beneath it is all of the temperatures and the times. It would be worth it to, I want to say memorize them because I don't know what's going to be on the final, but I don't want to make you guys do unnecessary studying. But I feel it would be worth it for you guys to memorize those if you haven't already. And I have the answers for it underneath my slide, so feel free to like look back and forth between them if you would like. But using heat, you're going to either do autoclaving, pasteurization, UHT treated, or dry heat sterilization. And if you can kind of know the differences between those, then you should be good. Other ways to sterilize would be membrane filtration. So taking a tiny membrane that's got really, really tiny holes to separate something from something, that'll pull out the larger particles so that you can, and these are for like solutions that if you were to heat it up would kill whatever you needed in the end. So like let's say you've got a culture that has both fungus and bacteria in it and you only want the bacteria. You could essentially put a membrane there that had small enough holes in it for the bacteria to go through, but not small enough holes in it for the fungus to go through. So there's that one. Plus we have high pressures. So with high pressures, what's going to happen is it's like those hydraulic press videos almost, where you like squish something. But in this case, you're going to have it in like almost a vacuum chamber, and just put a lot of like pressure in there through the atmosphere, which will cause it to just lyse. It'll cause it to like break apart like an Orbeez or Orby. Like, yeah, anyways, it gets squished by just the atmospheric pressure. Then we've got radiation, which we talked about with the ionizing versus non-ionizing. I spelled that wrong. Ionizing. Okay. With the ionizing versus non-ionizing and how one of them will break the DNA, whereas the other one will form those thymine dimers. So there's that. She didn't really talk about DNA replication. I have some DNA replication stuff in here, but every time I tried to talk to you guys about it in class, it seemed that she hadn't really gone over it. If she did, I will send out a video to help you guys out with that, but I'm not going to go over it because I really don't think she went over it too much, because I don't think she went over it too much. Just kidding. I have a video linked in here if you guys want it. It should be one of those crash course by Tom Green, Hank Green, and I love it. It's probably one of the best ones he's made. So there's that. One thing I do want to talk about is eukaryotic cells have introns and exons. Exons are going to be what's expressed. Introns are in the way. So when you're making a protein and starting to form your genetic code, all those introns have to be removed and exons have to be spliced together before it can leave the nucleus. She's talked about it in multiple semesters, and I feel like she didn't talk about it in this semester. But that, I mean, if she didn't talk about it, more knowledge to you. I think it's one of the cooler things about DNA replication and protein synthesis. But there is that. She did talk about mutations. I know we talked about this one. So they can be beneficial, disadvantageous, or silent. With silent, basically what happens is it's going to change out a nucleotide, but once the nucleotide is able to be coded in the codon during protein synthesis, it'll code for the same amino acid. I don't have any specific examples because I don't have a genetic code right in front of me, but it just means that the amino acid in the end is going to be the exact same as it was in the beginning. Then we have a missense protein, a missense mutation, and this results in an amino acid substitution. So just like in silent mutation where you have one nucleotide that changes, what's going to happen here is that nucleotide is going to change, but it's going to code for a completely different amino acid, meaning that if your protein was calling for arginine and you got leucine instead, it would just code for a different amino acid, which could essentially affect the protein's structure. It just kind of depends. Sometimes it doesn't. Then we've got a nonsense protein, a nonsense mutation, which will code for a premature stop codon. So our stop codons, we've got lots of them, but it's going to code for a premature stop codon, meaning you could have 2 amino acids and then that stop codon, and then the next 5 amino acids that should have been in that protein just aren't there anymore. There's that. And then frame shift is going to result from either an insertion or a deletion. So you're going to either have one nucleotide missing or one nucleotide that's going to be inserted, which just causes it to shift off by one. And on the slide after this one, it should be slide 34, it kind of shows you all of them, every single one of the mutations on there. So that's how that one works. It's just gonna kind of shift it off. It's like when your zipper is kind of messed up and there's one tooth that's sticking out and then it gets stuck because that one tooth is sticking out. It's kind of weird, but that's the best way I can describe that one. And then the last thing I wanted to talk about from these chapters, at least, is PCR. So it's going to use Taq polymerase. And in parentheses, I ask why. I do have the answer below. I don't want to tell it to you. I want you guys to think about it first. But PCR is going to ask us if there is evidence of bacterial DNA. And it's often used in crime analysis to replicate DNA really, really fast because it can do that. But one distinction I want to make, because I don't know if I make it on my next set of slides that I'm going to be going over this Saturday, is this one is looking for evidence of DNA, whereas ELISA and Western blot testing is going to look for evidence of seroconversion. So it's going to look for antibodies instead of actual genetic material. But I think that is it. You made it through an hour of the first 10 chapters. When I made these slides, I kind of pulled from what I thought was most important and what I thought were the concepts that most people struggle with. So I think it's worth it to do your own studying outside of it. I don't want my reviews to be the only thing you study from, because they're not entirely complete, and the way that I teach may not suit the way that you learn. So please feel free to message me if you have any questions, and I'll have both slides sent out to you guys shortly after this video is, if not before this video is. And I can't wait for you guys to take your final. I know you guys feel differently, but I think you're going to do amazing. So let me know if you have any questions, and happy studying.